Hi guys, this is Miss Michael, and this is a short video on gene therapy. And I'm going to use this YouTube video because it has great graphics, but I'm going to pause it intermittently to explain what's going on with the diagrams.
Develop
So,
long-lasting therapies for people with severe diseases. Our initial focus is in
so first of all, let's define what gene therapy is. Gene therapy is um, a biotechnological process where um, doctors are able to isolate a gene that has a mutation, and they're able to insert a correction using DNA. So instead of treating a disease with medications or other therapeutics, what they will do is they will isolate the gene that is causing the disease or the problem, and they will put a correct code into a viral vector or into some stem cells and deliver the correction to the cells at that level. And then over time, as mitosis of those cells generates more cells, it creates a situation where the mistake has been corrected at the level of the genome. Okay.
ophthalmology. Genetic defects in over 200 known genes have been associated with poor visual function. Genes are segments of DNA that direct the cell to produce proteins that perform a vast array of functions in the body.
So here it's showing a strand of DNA and that little sliver, that section of the gene. And as you learned in the first semester with protein synthesis, that DNA has the code, and the code is transcribed into RNA, and the RNA is translated into a string of amino acids that are folded up to look like the large mass that you see on the screen, the protein.
Many diseases have a genetic basis, meaning that mutated genes provide incorrect instructions that cause the cell to produce an abnormal protein.
Okay, so a DNA that has a mutated gene, again, it's going to make an abnormal protein. Some examples of abnormal proteins would be the um, the hemoglobin protein in sickle cell anemia. It's not shaped; it makes it so it's malformed, and it causes a lot of problems in the body. Cystic fibrosis is a disease where there are certain ion channels in side of the cell membrane, and those channels are proteins, and the proteins are abnormal because they come from incorrect code within the DNA.
Or disable production of a protein completely, either of which can cause disease. In gene therapy, a functional copy of the gene is delivered into a patient's own cells. Normal protein that is produced from a functional gene has the potential to correct the underlying cause of the disease and provide restorative therapeutic benefit. Gene therapy uses engineered viruses to deliver genes into cells.
So, an engineered virus, that is a viral vector. And what they do is they take viruses that attack certain cells, and they take out the genetic information within the virus. That's the truly harmful part of the virus. And they use that virus as a vesicle to insert the DNA, because DNA is very delicate without a protective coating. So they need some sort of mode for that DNA to deliver the correction to the cells.
These are called viral vectors. Vectors made from adeno-associated virus, or AAV, are especially well suited for treating retinal diseases. AAV is a small, simple virus that elicits only a weak immune response and has never been shown to cause disease in humans. In gene therapy, the two native AAV genes are removed and replaced with DNA that contains a functional gene with instructions to produce a normal protein. The DNA also contains a promoter, which drives production of the functional protein. The promoter and gene are flanked by signals called inverted terminal repeats that allow the DNA to be packaged inside the protein shell of the AAV vector. AAV vectors are delivered to cells in the retina by routine ophthalmic delivery methods, such as intravitreal or subretinal injection. Once administered, AAV vectors enter the patient's cells and deliver a copy of the functional gene directly into the nucleus. Once inside the nucleus, the functional gene can support the production of the normal protein and correct the underlying cause of the disease.
Okay, so in this video, they're showing a particular mutation that has to do with sight. And so they're using the viral vector. In some instances of stem cell, excuse me, in gene therapy, there's also the use of stem cells. And stem cells are cells that can be programmed. They don't have a specific function, and they can be used also to deliver the correction. Again, gene therapy is a way that um, doctors can deliver a corrective section of DNA that changes a protein from a non-functional protein to a healthy protein.